Good evening. Am I audible and visible? If I'm audible and visible, can I get a quick thumbs up, please? Good evening, Dr. Sarika. Good evening, Deepti. So, if you're joining the session for the first time, I am Dr. Shanmugapriya, you're an academy educator for biochemistry. And today we'll be discussing few PYQs, previous year questions in NEET PG and INI CET as far as biochemistry is concerned. And using those MCQs, we'll try to revise and recall relevant facts and concepts. Okay, so I will not only be discussing those MCQs, I will also be giving you relevant facts and concepts, so that by the end of the session, you would have understood at least like 50 concepts and few facts. Okay. Good evening, Moon Krish. So hi everyone. Before we start the session, let me tell you a few facts about Unacademy's plans. So first, about the Republic Day celebration offer, you have 20% off on all medical PG subscription plans. Good evening, Moon Krish. Yes. So this is valid till tonight 11:59 PM. This is valid. Wherein if you want a 36-month subscription, all that you will have to pay is 35,112. For 12-month subscription, you will have to pay just 21,183. And to subscribe to this, you can use any educator's code. You can use my code, which is C Shanmugapriya, my name. Okay. So that's about the Republic Day celebration. And then if you have any of these Unacademy NEET PG subscription plans, then you will have an access to the final booster batch. And this final booster batch is starting just tomorrow. And this includes 3 modules. The first module will include 28 days of rigorous previous year question discussion. All 19 subjects will be discussed. And module 2 will include most expected questions. And module 3 will include BTR series by Dr. Zainab. And please do not forget to attend this All India mock test, which is on Feb 5th at 9 AM. So that's all about Unacademy's plans. Let's start discussing these MCQs one by one. This is a very interesting MCQ. Primaquine can precipitate hemolytic anemia in individuals with, good evening Shruzana, hi Ghost Rider. So primaquine can precipitate hemolytic anemia in individuals with an enzyme deficiency. The enzyme is related to which pathway? Good Sarika, good Tarun, yes. All of you very good. Deepti, Moon, Deva, yes. It is related to which pathway? It is related to HMP shunt. Okay. So let me tell you, let me summarize this question. The question history tells you that following exposure to oxidative stress. You know, primaquine is a source of oxidative stress. Following exposure to oxidative stress, there is an individual who is presenting with hemolytic anemia. Okay, and as far as oxidative stress is concerned, do you all know that RBCs act as a major source of oxidative stress in your body? Yes, right? Because you know RBCs carry hemoglobin. No, RBCs carry oxygen, and as and when they carry oxygen, what they tend to do is they start giving out electron. The Fe2 plus has got a propensity to give out its electron to oxygen, converting oxygen to superoxide radical. And you know, from this superoxide radical, you get hydrogen peroxide that becomes peroxyl radical, and that initiates oxidative stress. So in your body, which is the major source of oxidative stress, it is RBCs. Which means for RBCs, you need a special antioxidant mechanism. And that special antioxidant mechanism that is present in RBCs is called as glutathione. So can you tell me, how does glutathione help as an antioxidant? I am going to write down everything. Please have a look at it. So how does glutathione act as an antioxidant? Suppose an oxidative species like hydrogen peroxide gets generated in RBCs. 2 molecules of glutathione will react with this hydrogen peroxide. Both these molecules of glutathione will donate 1 hydrogen atom each. So what will happen to H2O2? H2O2 becomes H4O2. What is H4O2? It is nothing but 2 molecules of water. That way, can you see that you have detoxified hydrogen peroxide to non-toxic water? But in this process, what has happened to glutathione, all of you? Yeah, in this process, what has happened to glutathione? Very good, Deepti. Glutathione has got oxidized to form GSSG. And if I ask you to name this enzyme, can we call it as a peroxidase? Why will you call it as a peroxidase? Because using this enzyme, you have detoxified hydrogen peroxide, so you call it as a peroxidase. And you have used glutathione for this purpose, so the enzyme is glutathione peroxidase, which helps in detoxifying hydrogen peroxide. With that, can you rest? Do you think your RBCs can rest after detoxifying hydrogen peroxide? They cannot. Because what are they supposed to do? They are supposed to regenerate glutathione. How will you be able to regenerate glutathione? By reducing this glutathione, which is oxidized. So the enzyme that you want is glutathione reductase. And this glutathione reductase needs NADPH. This glutathione reductase needs NADPH. From where do you get this NADPH? You get this through HMP shunt. Do you all know that there is an enzyme in HMP shunt which is called as glucose 6-phosphate dehydrogenase? Do not get confused between glucose 6-phosphatase and glucose 6-phosphate dehydrogenase. Glucose 6-phosphatase is an enzyme of glycogenolysis and gluconeogenesis. Whereas glucose 6-phosphate dehydrogenase is an enzyme of HMP shunt which will act as a source of NADPH. So can you tell me what is expected whenever there is a defect of HMP shunt? Without glucose 6-phosphate dehydrogenase, NADPH will not be generated. So glutathione cannot be regenerated. So RBCs are missing an effective antioxidant mechanism. Without an effective antioxidant mechanism, RBCs will become susceptible to lysis following exposure to an oxidative stress. So if the history says that there is a child or a person who is presenting with hemolytic anemia following exposure to an oxidative stress, immediately that should ring a bell in your head. What should you think of? You should think of HMP shunt defect. It should be G6PD deficiency. Is that clear? So to summarize it, there is a patient who is presenting with hemolytic anemia following exposure to oxidative stress, which means there is a defect of regenerating glutathione, which means there is low NADPH. When will there be, is my voice low? Is my voice low? Audio is not good. Am I audible now? Am I audible now? Give me a second. Is it good now? Is it good now? Okay, good. So I'm happy that's fine now. So that's about, so NADPH is low, which means it's a defect of which pathway? It means it's a defect of HMP shunt. I'm happy that's fine now. So, okay. So what are the other facts that can be asked to you as far as this question is concerned? What are the relevant facts that you need to know? They can ask you what are the sources of NADPH. So can you tell me what are the sources of NADPH? The major source of NADPH is HMP shunt. And apart from HMP shunt, it is also cytoplasmic isocitrate dehydrogenase. The second one is cytoplasmic isocitrate dehydrogenase. And the third one is malic enzyme which converts malate to pyruvate. This every one of you should know by heart. Anytime you are asked, you should be able to answer this. What are the sources of NADPH? Number one is HMP shunt. Number two is cytoplasmic isocitrate dehydrogenase. And number three is malic enzyme which converts malate to pyruvate. And what are the other facts that you need to know? Double voice and echo. Is there an echo now? Is there an echo? No, right? Okay. Hippo, probably you'll have to refresh something from your side or check your speaker. Okay. So what are the other facts that you need to know about HMP shunt? Few facts about HMP shunt. So there are two phases in HMP shunt. One is an oxidative phase. The other one is a non-oxidative phase. An oxidative phase is otherwise called as a completely irreversible phase. Okay, whereas non-oxidative phase is otherwise called as a reversible phase. Now, what is the significance of the oxidative phase or irreversible phase? That everybody knows, right? In this step, you oxidize glucose 6-phosphate. You remove hydrogen from glucose 6-phosphate. You give that hydrogen to NADP, forming NADPH. So, what is the one significance of the oxidative phase? It acts as a source of NADPH. Whereas, what is the significance of the non-oxidative phase or reversible phase? This acts as a source of ribose 5-phosphate, and that is why you call HMP shunt otherwise as PPP pathway. Why is it called as PP pathway? It is pentose phosphate pathway because it acts as a source of a pentose phosphate, which is ribose 5-phosphate. Now tell me, what are the uses of NADPH? Okay, good. Hippos. Hippos are idiots. Okay, from where did you get this name? Hippos are idiots. Okay, that's an interesting name to have. So tell me, what are the uses of NADPH? The major use of NADPH, just now I told you, it is necessary as a coenzyme for what? It's necessary as a coenzyme for glutathione reductase, which is involved in regenerating glutathione, which is an important antioxidant mechanism of RBCs. It helps in regenerating glutathione, which is an important antioxidant mechanism of RBCs. The second use of NADPH is it is necessary for reductive biosynthesis of all the lipids. So I hope you all know that lipids are more reduced, right? Hippos has got a good fun here. So if you get time, please type why you call yourself as hippos are idiots. Okay. So lipid biosynthesis. Do you all know that lipids are more reduced than carbohydrates? You know, lipids give you more energy than carbohydrate. That by itself tells you that lipids are more reduced. So whenever you are trying to synthesize lipid, there will be multiple reduction steps. And for all these reduction steps, you need a hydrogen source. What will act as a hydrogen source? It is NADPH. Yeah, it is NADPH. So after this session, you go back, check any textbook that you have a look at the pathway of lipid synthesis, be it fatty acid synthesis or cholesterol synthesis or bile acid synthesis. In all these synthetic pathways, you will be using NADPH. So it is necessary for reductive biosynthesis of all the lipids. Next use is NADPH is necessary as a coenzyme for ribonucleotide reductase. So if you are trying to convert nucleotide triphosphates to deoxynucleotide triphosphates, you need ribonucleotide reductase. And this enzyme uses NADPH. And the last, I won't say this is the last use, the probable question can be related to NADPH. This was asked in the latest FMG. They asked you cytochrome enzymes use what as a coenzyme? All cytochrome P450 enzymes use NADPH. So it is also used by cytochrome P450 enzymes. Okay. Hippos you think are idiots. Okay, I'm sorry Mota Babi, I forgot that you asked about electron transport chain, but I'll definitely help you learn about electron transport chain during this session. Okay. Before I end the session, I will tell you. Okay. So that's about the uses of NADPH. So please remember, cytochrome enzymes use what as a coenzyme? NADPH as a coenzyme. Now what is the use of ribose 5-phosphate? Ribose 5-phosphate is like a foundation based on which you build, hi Umesh Patel. So ribose 5-phosphate is like a foundation based on which you build all nucleotides. To ribose 5-phosphate, if you attach adenine, it is called as AMP. You attach guanine, it is GMP. So it is necessary for purine and pyrimidine nucleotide synthesis. Okay. So that's about the relevant facts and concepts as far as this MCQ is concerned. So tell me, what is the right answer here? Yes, Mota Babi, I said I will teach you. I will teach you. Okay. So what is the right answer here? The right answer here is HMP shunt. So answer this: on prolonged starvation, which of the following tests will be positive? Prolonged starvation, do you know starvation causes ketosis? They are asking you which of these tests is a test done to detect ketone bodies. Which is the test done to detect ketone bodies? It is Rothera's test. Your free YouTube live classes help me revise the subject. You're awesome. Thank you, Sahadev. I'm happy that you are liking it. I'm happy that I'm able to help you. So Rothera's test. So whenever you see a purple colored ring, whenever you see a purple colored ring, it is Rothera's test, which is a test done to detect ketone bodies. Okay. So I'm going to help you understand all these tests. So what do you think is test A? Test A, when you see an yellow colored solution. Yellow colored solution is most probably xanthoprotic acid test. Have you all heard of xanthoprotic acid test? Even if you don't remember the name, do you remember you working in the laboratory with dilute nitric acid? And whenever dilute nitric acid spills in your palms, you must have observed yellowish discoloration of the palm. And that is because of xanthoprotic acid test. And it is because of nitric acid reacting with aromatic amino acids. And your sweat is rich in aromatic amino acids. So whenever dilute nitric acid spills on your sweat, that is an yellow discoloration. So that is xanthoprotic acid test done to detect the presence of aromatic amino acids. So can anybody tell me what are the aromatic amino acids? Yeah, just out of topic here. What are the aromatic amino acids? Phenylalanine, tyrosine and tryptophan. Okay, phenylalanine, tyrosine, tryptophan are the three aromatic amino acids. So can you tell me what is this test wherein blue color becomes red? Very good, Sarika. Excellent, Moon Krish. Good, Sapna. Very good. So tell me all of you, what is this blue color becoming red? That is Benedict's test. And what is the purpose of Benedict's test? Very good, Dr. Dreamer. Very good, Dr. Sarika. Again, good, Mayank. Good, Deepti. So Benedict's test is done to detect the presence of a reducing substance in a given solution. It is not just reducing sugar. Any reducing substance will be answered by Benedict's test. And what is C? This is what we have been discussing, right? C is Rothera's test, which is done to detect ketone bodies. And what is this? Yellow color solution becoming dark green or black. Yeah, yellow color solution becoming dark green or black. There are two possibilities. One is urine turning dark on standing. In which condition do you think urine turns dark on standing? That is called as alkaptonuria. What does alkapton means? Alkapton means black. Alkaptonuria is black urine disease. That is one possibility. Another possibility is it can be benzidine test. So even in benzidine test, if the urine has got blood, it will answer benzidine test with a dark green or black precipitate. Very good. Yes, it can be alkaptonuria or it can be benzidine test, which is answered by blood and urine. Okay. So I'm going to quickly tell you about all the color reactions that can be used to detect an abnormal substance in the urine. Okay. So I want your help in filling up this tabular column. All of you start answering. So the first abnormal constituent that we are all interested about is reducing substance. So when I say reducing substance, it can be glucose, which is found in diabetes. It can be fructose, which is found in fructose intolerance. It can be galactose in galactosemia, or lactose, which is found in pregnancy or lactation. Okay, so any of these reducing substances, if it is present in urine, will answer which test? Very good. Will answer Benedict's test. And in Benedict's test, what is the observation? Observation will be the blue color Benedict solution will slowly become green or yellow or orange or red. And you know the color will depend upon the concentration of the reducing substance that is present in the given solution. Okay, so that's about Benedict's test done to detect the presence of a reducing substance. Now the next one is ketone bodies. Tell me, what is a test done to detect ketone bodies? It is Rothera's test. And in Rothera's test, what is the observation that you see? The observation is a purple colored ring. And as far as Rothera's test is concerned, please remember Rothera's test will be answered only by acetoacetate and by acetone. What are the 3 ketone bodies, all of you? So this is more or less like a revision session, yeah. So I want you to recollect all relevant facts and concepts so that you are able to connect all the dots. Okay, so tell me what are the 3 ketone bodies. The first one is acetoacetate. The second one is acetone. The third one is beta hydroxybutyrate. Of the 3 ketone bodies, Rothera's test will be answered only by acetoacetate and by acetone, never by beta hydroxybutyrate. Got it? This can be asked as an MCQ. So that's about Rothera's test, which gives you a purple colored ring. Now after ketone bodies, protein. What is the test done to detect if there is protein in urine? To detect if there is protein in urine, the test is called the sulfosalicylic acid test. Yeah, the sulfosalicylic acid test, where you will see a white color precipitate. I have seen many students answering it as biuret test. Please remember, biuret test is a test done to detect protein in blood. If you want to detect protein in urine, you don't do biuret test, you perform sulfosalicylic acid test, when white color precipitate is seen. Okay. So after protein, blood. So tell me which is the test done to detect blood in urine. Blood, B for B, benzidine test. And what will be the observation? The observation is, you know, the urine will form a dark green precipitate. Many a times it will look like a black precipitate. That is blood in urine detected by benzidine test with dark green or black precipitate. Okay. Now what is the test that is done to detect bile salts? Bile salts, do you remember playing with sulfur powder in your first year? Yeah, sprinkling the sulfur powder and checking whether the sulfur powder sinks to the bottom or not, right? That is Hayes test. And Hayes test is answered by bile salts, wherein sulfur powder sinks to the bottom. If sulfur powder sinks to the bottom, it is positive. It is because bile salts, what are bile salts? They reduce the surface tension of the urine. So if the given urine sample has got bile salt, it reduces the surface tension of the urine, so that when you drop sulfur powder dips to the bottom of the tube. Okay, that's about bile salt. Now what is the test that is done to detect, Dr. Dreamer, bile pigments? Observations are hidden by me. Okay, so you can get the PDF after the session, because I cannot go out of it. Okay. So after the session you can download the PDF. For now, listen to this and try to memorize. Okay. Dr. Dreamer, bile pigments will be detected by Fauchett's test. Okay. What will you observe in Fauchett's test? In Fauchett's test, you see a green precipitate. Blue or green precipitate on the filter paper. Okay. Blue or green precipitate on the filter paper is the observation that is seen in Fauchett's test that is done to detect bile pigments. So Fauchett's, Sarika, Fauchett's is not for bile acids. Okay, what are bile acids? Bile acids will become bile salts. How many of you have a confusion about bile salts and bile pigments? Yeah, how many of you have a confusion between bile salts and bile pigments? What are bile salts? Bile salts are salts of bile acids. And what are bile acids? Bile acids are derivatives of cholesterol. You understand this? Bile acids are derivatives of cholesterol. So cholesterol undergoes hydroxylation to form bile acids. Bile acids will become bile salts. And the use of bile salt is that they help in emulsification of the fat and they help in lipid digestion and absorption. Whereas bile pigments, what are bile pigments? These are bilirubin and biliverdin. Bilirubin and biliverdin, they are just excretory products of heme catabolism. They don't have any physiological role to play like bile salts. Do you understand this? You need bile salts because they help in lipid digestion absorption, whereas bile pigments, they are just excretory products of heme catabolism. Bile pigments will include bilirubin and biliverdin. Bilirubin and biliverdin is what you detect using a Fauchett's test. Bile salts are detected using Hayes test. Got it? Okay. So, how many of you have been wanting to know about receptors? Chemical messengers and receptors, right? This is being frequently asked. Sometimes they ask you vasopressin acts through which receptor? All of the following are G protein coupled receptors except. All of the following are intranuclear receptors except. Which of the following is an intracytoplasmic receptor? So for you to understand and answer about all these MCQs, I'm going to tell you all about receptors now. So let me give you a prelude. It will take some time. Yeah, it will take at least 5 to 10 minutes for me to tell you about all the receptors, but I want you to hang on and understand and memorize about all these receptors. Okay. So what is the right answer here? Which of the following chemical messenger utilizes intras? Very good, Shifana. Very good, Moon. Excellent. Very good, Dr. Dreamer. The answer is thyroxine, because thyroxine uses nuclear receptors. Hyperlipoproteinemias. Okay, I will tell you about hyperlipoproteinemias after this MCQ. Okay. So thyroxine uses nuclear receptors. Okay, it's an intracytoplasmic receptor. So tell me, how do you classify receptors? You classify receptors as surface receptors and intracellular receptors. And the choice of whether a chemical messenger uses a surface receptor or an intracellular receptor will depend upon the chemical nature of the substance. If the substance is a steroid, if the messenger is a steroid, it can cross any membrane. So it uses an intracellular receptor. That is why intracellular receptors are otherwise called the steroid receptor superfamily. Intracellular receptors are otherwise called the steroid receptor superfamily, and these steroid receptors can be either in cytoplasm or in nucleus. So let me tell you what are the nuclear receptors, which means remaining steroid hormones will utilize cytoplasmic receptors. So what are the nuclear receptors? Start from vitamin A receptor, RARRXR. Okay, vitamin A receptor, RARRXR. Vitamin D receptor, thyroxine. Okay, so RARRXR, vitamin D, thyroxine, all these are present in the nucleus, which means remaining steroid hormones have their receptors in the cytoplasm. So what are the remaining steroid hormones? You can start from glucocorticoid, mineralocorticoid, androgen, estrogen. So all these have cytoplasmic receptors. Got it? So that's about intracellular receptors. Do you have any doubt about intracellular receptors or steroid receptors? Vitamin A, vitamin D, thyroxine, all these are present in the nucleus. So now about surface receptors. If the chemical messenger is a simple protein, or if it's a glycoprotein, or if it's a polypeptide, it cannot cross a cell membrane. So it ends up using a surface receptor or a membrane receptor. This surface or membrane receptor will be of two types. One is ionotropic receptor. The other one is metabotropic receptors. So what are ionotropic receptors? These are ligand-gated ion channels. Ionotropic receptors are ligand-gated ion channels in the sense, whenever the ligand or the chemical messenger comes and binds to these receptors serve to open or close an ion channel. When they open or close an ion channel, they change the electrical activity of a cell and they bring about changes within the cell. So what are ionotropic receptors? Ionotropic receptors are nothing but ligand-gated ion channels. When the ligand or the chemical messenger binds to these receptors, the receptors will serve to open or close ion channels. So can you tell me what are the examples of Mota Babi? Can I tell you something? You cannot be in a hurry when you want something to be long lasting. Yeah, you cannot be in a hurry when you want something to be long lasting. It would not have taken me half an hour for me to complete like 5 to 10 MCQs. Okay, but that's not the aim of all these sessions. The aim of all these sessions to get all these concepts straight into your mind so that anytime these questions are asked, you will be able to answer it. Instead, if you are in a hurry today, you will not be able to recollect any of these facts in your exams. Okay, so I request all the students who are watching this session to spend time in understanding concepts and in memorizing facts. Okay, do not be in a hurry for something which has to be long lasting. Okay, I understand that Mota Babi says thoda jaldi padaiye and cover many topics. Yeah, it's not about covering many topics. If you cover many topics, you will not be able to answer even one MCQ. Okay. Yes. So what are the ionotropic receptors? Ionotropic receptors example. No problem. No problem, Steel. I understand that there are some students who are anxious about covering many topics. I do understand that also, but that does not work. Okay. So coming back to this ionotropic receptors, I told you acetylcholine. So I hope you all remember that acetylcholine binds to nicotinic receptors. When it goes and binds to nicotinic receptors, it opens up sodium channels. Don't we know that? So acetylcholine nicotinic receptors are examples for ionotropic receptors. And not only acetylcholine, glutamate. Glutamate has got both ionotropic and metabotropic receptors. Okay, so every teacher has got her own way of teaching, Mota Babi. This is how I teach. Okay. So glutamate has got both ionotropic and metabotropic receptors. Glutamate's ionotropic receptors will include kainate, AMPA and NMDA receptors. Okay, so what are the ionotropic receptors of glutamate? Kainate receptors, AMPA receptors and NMDA receptors. So kainate receptors has got Na, that tells you it opens up sodium channels. When glutamate binds to AMPA receptors, it either opens sodium channel or it closes potassium channel. Yeah, AMPA receptors either open sodium channel or it closes potassium channel. NMDA receptors will open up calcium channels. Do you understand this? So these are the examples of ionotropic receptors. So have a look at this. What are the examples of ionotropic receptors? All ionotropic receptors are ligand-gated ion channels. Example will include acetylcholine binding on nicotinic receptors which open up sodium channels. Yes, very good, Dr. Dreema. GABA A opens up chloride channel. That is also an example. Glutamate, AMPA, kainate and NMDA receptors. And GABA A channels, GABA A receptors open up chloride channels. Okay, so that's all about ionotropic receptors. Now about metabotropic receptors. If you look at metabotropic receptors, they are of two types. One is G protein coupled receptor, the other one is non-G protein coupled receptors. Okay, G protein coupled receptors and non-G protein coupled receptors. So first about G protein coupled receptors, as the name indicates, these are the receptors which are coupled to G proteins. So where will you have all these receptors? All these receptors are present in the cell membrane. And these receptors are coupled to G proteins. What are G proteins? These are proteins which get activated by binding to GTP. G protein is any protein which gets activated on binding to GTP. And there are two types of G proteins, small G proteins and heterotrimeric G proteins. And all G protein coupled receptors have got nothing to do with small G proteins. All your G protein coupled receptors are coupled only to heterotrimeric G proteins with alpha, beta, gamma subunits. Do you all remember this? Yeah. Okay, Motababi, I totally understand your concern. I will try to be quick. Okay. I totally understand your concern. Okay. Yeah. But if you want to know about receptors, it takes its own time, right? So you cannot just like that memorize what are the GS receptors, GI, GQ, GT. Tomorrow you're going to forget it. So just spend some time, just spend 5 to 10 minutes and learn all this so that it is there in your memory forever. Okay? I totally get it. You don't have to be sorry. Yeah, I know it's no hard feelings. Yeah. So heterotrimeric G protein has got alpha, beta, gamma subunits. Okay. So this is about the G protein. Now about the receptor. These receptors traverse the cell membrane 7 times. Yeah, all G protein coupled receptors traverse the cell membrane 7 times. So you call them as serpentine receptors. They are called as serpentine receptors. And any protein will have an amino terminal end and a carboxy terminal end. Amino terminal end faces the extracellular side and carboxy terminal end faces the cytoplasm. C for C, carboxy terminal end faces the cytoplasm. And to this carboxy terminal end is attached the heterotrimeric G protein with alpha, beta, gamma subunits. And usually there is a ligand binding domain on the third transmembrane domain. There is a ligand binding domain on the third transmembrane domain. As long as there is no ligand on the ligand binding domain, this alpha subunit is attached to GDP. Once the ligand comes and binds to the ligand binding domain, GDP is replaced by GTP. And once it is replaced by GTP, the alpha subunit will get detached from the beta gamma subunit. Once alpha subunit is attached to GTP, it gets detached from beta gamma subunit. After it gets detached from beta gamma subunit, depending upon what enzymatic activity this alpha subunit exhibits, G protein coupled receptors are of 4 types. On what basis do you classify G protein coupled receptors? Depending upon the enzymatic activity that the alpha subunit exhibits after dissociating from beta gamma. So what are the 4 types? GS is the first type. GI, we have GS, GI, GQ and GT type. Okay. So what does S here stand for? In GS type, the alpha subunit stimulates adenylyl cyclase. In GS type, the alpha subunit stimulates adenylyl cyclase. Cyclic AMP is produced that activates protein kinase A. And this protein kinase A phosphorylates and activates intracellular proteins. Example for GS coupled receptors. I hope you all know that glucagon acts through GS receptor. So I am going to tell you all the examples of GS coupled receptors. Please answer it along with me. Please try to memorize it during the session. Okay. So what are the GS coupled receptors? You have glucagon number 1. All beta adrenergic receptors. Okay. Glucagon, all beta adrenergic receptors. And then vasopressins B2 receptors. Do you all know that vasopressin acts through both B2 receptor and B1A receptor? Do you know that? Vasopressin acts through both B2 and B1A receptor. Vasopressin B2 is GS coupled. And then all dopaminergic receptors except D2. Yeah, all dopaminergic receptors except D2. Parathormone, ACTH. Okay, parathormone, ACTH. CRH, corticotropin releasing hormone. And then few glycoprotein hormones like FSH, LH, HCG. Okay, FSH, LH, HCG, all these are GS coupled receptors. So can you tell me what are the GS coupled receptors? Quickly tell me, can the chat box be made active please? So the GS coupled receptors include starting from glucagon, right? Glucagon, all beta adrenergic receptors. Vasopressins B2 receptor, all dopaminergic receptors except D2. And then you have parathormone, ACTH, corticotropin releasing hormone. And then few glycoprotein hormones which will include FSH, LH and HCG. Okay, so that's all about GS coupled receptors. Now what are the GI coupled receptors? Very good all of you. I'm happy that you're all answering. So what are the examples of GI coupled receptors? In GI type, the alpha subunit inhibits adenylyl cyclase. So what is decreased? Cyclic AMP is decreased. An example for all GI coupled receptors will be all inhibitory receptors. So can you tell me what are the inhibitory receptors? Don't you know that alpha 2 decreases central sympathetic outflow? Yeah, alpha 2 decreases central sympathetic outflow. Alpha 2 is GI coupled. And you know M2. Yeah, M2 is present in heart. Right, M2 is present in heart and vagal stimulation depresses heart. So M2 is inhibitory, it is GI coupled. I said all dopaminergic receptors except D2 or GS coupled because D2 is GI coupled. So tell me what are the GI coupled receptors? Alpha 2, M2 and D2 receptors. Okay. Now what is GQ type? In GQ type, the alpha subunit stimulates phospholipase C. In GQ type, the alpha subunit stimulates phospholipase C. This phospholipase C acts on phosphatidylinositol diphosphate, converts PIP2 to IP3 and DAG. Phospholipase C acts on phosphatidylinositol diphosphate, converts it into IP3 and DAG. IP3 causes calcium release from the storehouses and DAG activates protein kinase C. Okay, IP3 causes calcium release from storehouses, DAG activates protein kinase C. So what are the GQ receptors? everything in one. Yeah, GQ will mostly include ones. So what are the ones? As far as adrenergic receptors are concerned, I said beta adrenergic receptors are all GS coupled. Alpha 2 is GI coupled. So what is left out? Alpha 1. Right? So alpha 1, M1, M3. Yeah, alpha 1, M1, M3, B1A receptor. Okay, alpha 1, M1, M3, B1A receptor, angiotensin receptors. Okay, and apart from angiotensin receptor, please memorize GnRH. Okay, you can have a mnemonic for the remaining chemical messengers. It is GOVA. So what does GOVA stand for? GnRH, oxytocin and angiotensin 2 and angiotensin 2. Have you understood this? So tell me, what are the GQ coupled receptors? All 1 receptors. What are the 1 receptors? Alpha 1, M1, M3, B1A receptor. Right? Alpha 1, M1, M3, B1A receptor. Apart from this, it is GOVA. What does GOVA stand for? GnRH, oxytocin and angiotensin 2. All these are GQ coupled. Okay. Now GT type. In GT type, the alpha subunit stimulates phosphodiesterase specific for cyclic GMP, and it decreases cyclic GMP levels, and it is present in rods and cones. GT type is present in rods and cones. Okay, so are you clear about all G protein coupled receptors? Will you have any confusion here? So can I ask you a few questions? Will you be able to answer that in the chat box, please? Yeah. So tell me, how about oxytocin? Oxytocin acts through which receptor? Oxytocin comes under which category? GOVA. Where did I tell you GOVA? GQ type. Okay, oxytocin is GQ type. Okay, where will you place alpha 1? All 1 receptors. GQ has got 1, right? Yeah, 1. So alpha 1, M1, M3, B1A. All these are GQ receptors. Tell me about D2, dopaminergic receptor D2. All 2 receptors are GI coupled. So GI coupled receptors include alpha 2, D2 and M2 receptors. Okay, so that's all about G protein coupled receptors. So now about non-G protein coupled receptors. This is the last category, non-G protein coupled receptors. These non-G protein coupled receptors will have some enzymatic activity within themselves. And there are 2 enzymatic activities which are possible. Your receptors can either have guanylyl cyclase activity or they can have tyrosine kinase activity. Now, what are the chemical messengers which have guanylyl cyclase activity? This has been asked so many times in NEET PG. Atrial natriuretic peptide and nitric oxide. Yeah, atrial natriuretic peptide and nitric oxide have guanylyl cyclase activity. Now, tyrosine kinase receptor, they are of 2 types. One is intrinsic tyrosine kinase receptor, the other one is JAK-STAT pathway. Okay, one is intrinsic tyrosine kinase, the other one is JAK-STAT pathway. So, where will you find intrinsic tyrosine kinase activity? This is the famous insulin receptor. I hope you all remember that insulin receptor has got intrinsic tyrosine kinase activity. Apart from insulin, epidermal growth factor and platelet derived growth factor. Okay, insulin, epidermal growth factor, platelet derived growth factor have intrinsic tyrosine kinase activity. JAK-STAT pathway is exhibited by growth hormone, prolactin and all the other cytokines. Okay, JAK-STAT pathway is exhibited by growth hormone, prolactin and all the other cytokines. Okay, so that's all about receptors. So can we summarize all that we have learned so far? Yeah, so I'm going to summarize. I'm going to tell you the outline of classification of all these receptors with your help. So tell me, what are the 2 types of receptors, whether they are present in membrane. If they are present in membrane, I will call it as a surface receptor or I will call it as an intracellular receptor. And what will you call, thank you Lakshay, I'm happy that you're liking it. What will you call intracellular receptors otherwise is, you will call them as steroid receptors. And if you take steroid receptors, where are they present? Steroid receptors can be present either in cytoplasm or in nucleus. So tell me, what are the nuclear receptors? Nuclear receptors include vitamin A receptor RAR, RXR, vitamin D receptor and thyroxine. Okay, the remaining steroid receptors are there in cytoplasm. So that's all about intracellular receptors. Now how will you classify surface receptors? Surface receptors can be of two types. One is ionotropic receptor, the other one is metabotropic receptors. And what are ionotropic receptors? These are ligand-gated ion channels. Can you give me examples for ionotropic receptors? What did I tell you? Acetylcholine binding on nicotinic receptors. Glutamate has got kainate, AMPA and NMDA receptors. And then GABA A receptors. What does GABA A receptor do? It opens up chloride channel. That's about ionotropic receptors. Okay. So now about metabotropic receptors. There are two types of metabotropic receptors. One is G protein coupled receptors. The other one is non-G protein coupled receptor. So how will you classify G protein coupled receptors? GS, GI, GQ, GT type. Right? GS, GI, GQ, GT type. GS includes glucagon, beta adrenergic receptors, all dopaminergic receptors except D2, vasopressins B2 receptor, parathormone, ACTH, CRH. Right? ACTH, CRH, and then glycoprotein hormones, namely FSH, LH and HCG. Okay. GI, all inhibitory receptors, alpha 2, M2, D2. GQ in 1. What are the ones? Alpha 1, M1, M3, B1A. And then GOA. What does GOA stand for? GnRH, oxytocin, angiotensin 2. GT type is present in rods and cones. Now, non-G protein coupled receptors, they have enzymatic activity. There are two possible enzymatic activities. One is guanylate cyclase activity. The other one is tyrosine kinase activity. So where do you see guanylate cyclase activity? It is atrial natriuretic peptide or nitric oxide. Tyrosine kinase can be intrinsic tyrosine kinase or JAK-STAT. So where do you find intrinsic tyrosine kinase? The famous insulin, epidermal growth factor and platelet derived growth factor. JAK-STAT is seen in growth hormone and prolactin. Okay. So that's all about receptors. This is a summary of receptors. Okay. I hope it was of use. So answer this question. Which of the following chemical messengers utilizes intracellular receptors? Do you have any doubt now? Tell me about growth hormone. Growth hormone uses JAK-STAT. JAK-STAT is the mechanism. Where is it present? Growth hormone receptor is present in the membrane. It's a surface receptor. Right. Insulin is a tyrosine kinase receptor, intrinsic tyrosine kinase receptor that is also in the membrane. It is a non-G protein coupled receptor. Thyroxine is an intracellular receptor. To be very precise, it is in the nucleus. How about ACTH? Yeah, what did I tell you about ACTH, PTH, ACTH? All these are GS coupled. They are all G protein coupled receptors which are present in the membrane. Okay. So answer is thyroxine. Someone was asking about what? I think about lipoprotein, hyperlipoproteinemias, right? Let me tell you about that because there is a free space here. I'll use this free space. Okay. So hyperlipoproteinemias. You can classify hyperlipoproteinemias. Hippos asked. Okay. So you can classify hyperlipoproteinemias into three categories. Actually, how many types are there? Fredrickson's classification of hyperlipoproteinemias. There are 6 types. Type 1, 2A, 2B, 3, 4 and 5. How many types totally? 6 types. And these 6 types can be classified into 3 categories. The first category presents with only an elevation of triglyceride. The second category presents with an elevation of only cholesterol. The third category presents with an elevation of both. Okay, TGL elevation, cholesterol elevation, both elevation. Okay, so why should you classify it this way? Because based on this, clinical presentation will differ. If a patient presents with only an elevation of TGL, then the patient presents with eruptive xanthomas. Please memorize it. What do they present with? They present with eruptive xanthomas, recurrent pancreatitis. On examination, you will find lipemia retinalis. So tell me, what will be the observation when a patient presents with only an elevation of triglyceride? It is eruptive xanthomas, recurrent pancreatitis, and on examination you will find lipemia retinalis. Whereas if a patient presents with only an elevation of cholesterol, it will present as tendon xanthomas. Eruptive xanthomas and tendon xanthomas are totally different. Tendon xanthomas are small eruptive lesions which are found along the line of attachment of tendons. For example, along the knuckles or along the ankles, wherever tendons go and get attached, if you see small eruptive lesions, those are tendon xanthomas, which mean the person has got hypercholesterolemia. Whereas eruptive xanthomas are even tinier than tendon xanthomas. They are present along the extensor surfaces of your limbs. Yeah, they are present along the extensor surfaces of the limb, that is eruptive xanthoma. If you see a patient with eruptive xanthoma, that's a sign that the person has got elevated TGL. Okay, so they present with tendon xanthomas and they present with accelerated atherosclerosis. Okay, whereas if there is an elevation of both cholesterol and TGL, there will be a combination of both these manifestations. So because this classification will help you in diagnosis, will help you in managing the patient, I am asking you to classify it into 3 categories. Now what are the hyperlipoproteinemias which present with an elevation of only triglyceride? Elevation of only triglyceride is seen in type 1, type 4 and type 5. Only an elevation of TGL is seen in type 1, type 4 and type 5. Type 1 is otherwise called as familial chylomicronemia syndrome. Type 1 is otherwise called as familial chylomicronemia syndrome. And you know, chylomicron carries TGL. So if it's familial chylomicronemia syndrome, which lipid will be elevated? Triglyceride level will be elevated. Type 4 and type 5, they are called as familial hypertriglyceridemias. Okay, type 4 and type 5 are called as familial hypertriglyceridemias. Okay. Now, which hyperlipoproteinemia presents with only an elevation of cholesterol? That is type 2A. Only an elevation of cholesterol is observed in type 2A. That is why type 2A is otherwise called as familial hypercholesterolemia. Don't you think it's a logical name? Type 2A, there is only an elevation of cholesterol, so you call it as familial hypercholesterolemia. So can you tell me what are the mixed conditions? What is left out? Type 2B and type 3. Okay, type 2B and type 3 are the mixed conditions. So what is type 2B called as? Type 2B is called as familial combined hyperlipidemia. Again a logical name. Yes or no? Combined hyperlipidemia, to tell you that there is an elevation of both cholesterol and triglyceride. So type 2B is familial combined hyperlipidemia. Type 3 is otherwise called as remnant disease. Okay, type 3 is otherwise called as remnant disease because type 3 is caused by a defect of ApoE. 3 and E sound similar. Yes or no? 3 and E sound similar. So type 3 is an ApoE defect. It is otherwise called as remnant disease. It can also be called as familial dysbeta lipoproteinemia. Remnant disease is also called as familial dysbeta lipoproteinemia. So does that answer your question, hippos? Hippos are idiots? Yes, broad beta disease. You're right. So tell me, what are the hyperlipoproteinemias which present with an elevation of TGL alone? Elevation of TGL alone will be type 1, type 4 and type 5. Only an elevation of cholesterol is type 2A. Elevation of both will be type 2B and type 3. ApoE and Alzheimer's. So the normal, when I say ApoE, all of us have ApoE, okay? And the ApoE form that we have is E3. All of us have E3. If E3 is replaced by E2, if there is a mutation E3 gene, because of which instead of a person having E3 allele, if there is E2 allele, that is type 3 hyperlipoproteinemia. E2 is type 3 hyperlipoproteinemia. Instead of E3, if an individual has got E4, yeah, E4, that makes a person susceptible to develop Alzheimer's disease. So to find out if a patient has got a propensity to develop Alzheimer's disease, you can check for mutation causing ApoE4. Got it? Hippo's happy. That's a good phrase. Okay, so that's about hyperlipoproteinemia. That's about receptors. I have a session at 7 o'clock, so I have to leave. Okay, so I will meet you again sometime in the next week with another set of MCQs, with another set of topics. Okay, so thank you all. See you.